Lesson 10, Proteins. Proteins are one of the most abundant organic molecules in living systems and have the most diverse range of functions of all macromolecules. Proteins may be structural, regulatory, contractile, or protective. They may serve in transport, storage, or membranes. Or they may be toxins or enzymes. Each cell in a living system may contain thousands of proteins, each with a unique function. Their structures, like their functions, vary greatly. They are all, however, amino acid polymers arranged in a linear sequence. Enzymes, which living cells produce, are catalysts in biochemical reactions, like digestion, and are usually complex or conjugated proteins. Each enzyme is specific for the substrate, a reactant that binds to an enzyme, upon which it acts. The enzyme may help in breakdown, rearrangement, or synthesis reactions. We call enzymes that break down their substrates catabolic enzymes. Those that build more complex molecules from their substrates are anabolic enzymes, and enzymes that affect the rate of reaction are catalytic enzymes. Note that all enzymes increase the reaction rate and therefore are organic catalysts. An example of an enzyme is salivary amylase, which hydrolyzes its substrate amylose, a component of starch. Hormones are chemical signaling molecules, usually small proteins or steroids, secreted by endocrine cells that act to control or regulate specific physiological processes, including growth, development, metabolism, and reproduction. For example, insulin is a protein hormone that helps regulate the blood glucose level. Proteins have different shapes and molecular weights. Some proteins are globular in shape, whereas others are fibrous in nature. For example, hemoglobin is a globular protein, but collagen, located in our skin, is a fibrous protein. Protein shape is critical to its function, and many different types of chemical bonds maintain this shape. Changes in temperature, pH, and exposure to chemicals may lead to permanent changes in the protein's shape, leading to loss of function, or denaturation. Different arrangements of the same 20 types of amino acids comprise all proteins. 2 rare new amino acids were discovered recently, selenocysteine and pyrrolysine, and additional new discoveries may be added to the list. Amino acids are the monomers that comprise proteins. Each amino acid has the same fundamental structure, which consists of a central carbon atom, or the alpha carbon, bonded to an amino group, a carboxyl group, and to a hydrogen atom. Every amino acid also has another atom or group of atoms bonded to the central atom known as the R group. Scientists use the name amino acid because these acids contain both the amino group and carboxyl acid group in their basic structure. As we mentioned, there are 20 common amino acids present in proteins. 9 of these are essential amino acids in humans because the human body cannot produce them, and we obtain them from our diet. For each amino acid, the R group, or side chain, is different. The chemical nature of the side chain determines the amino acid's nature, that is, whether it is acidic, basic, polar, or nonpolar. For example, the amino acid glycine has a hydrogen atom as the R group. Amino acids such as valine, methionine, and alanine are nonpolar or hydrophobic in nature, while amino acids such as serine, threonine, and cysteine are polar and have hydrophilic side chains. The side chains of lysine and arginine are positively charged, and therefore these amino acids are also basic amino acids. Proline has an R group that is linked to the amino group, forming a ring-like structure. Proline is an exception to the amino acid's standard structure since its amino group is not separate from the side chain. A single uppercase letter or a 3-letter abbreviation represents amino acids. For example, the letter V, or the 3-letter symbol VAL, represent valine. Just as some fatty acids are essential to a diet, some amino acids also are necessary. These essential amino acids in humans include isoleucine, leucine, and cysteine. Essential amino acids refer to those necessary to build proteins in the body, but not those that the body produces. Which amino acids are essential varies from organism to organism. The sequence and the number of amino acids ultimately determine the protein's shape, size, and function. A covalent bond, or peptide bond, attaches to each amino acid, which a dehydration reaction forms. One amino acid's carboxyl group and the incoming amino acid's amino group combine, releasing a water molecule. The resulting bond is the peptide bond. The products that such linkages form are peptides. As more amino acids join this growing chain, the resulting chain is a polypeptide. Each polypeptide has a free amino group at one end. This end, the N-terminal, or the amino terminal, and the other end, has a free carboxyl group, also the C or carboxyl terminal. While the terms polypeptide and protein are sometimes used interchangeably, a polypeptide is technically a polymer of amino acids, whereas the term protein is used for a polypeptide or polypeptides that have combined, often have bound non-peptide prosthetic groups, has a distinct shape, and has a unique function. After protein synthesis, translation, most proteins are modified. These are known as post-translational modifications. They may undergo cleavage, phosphorylation, or may require adding other chemical groups. Only after these modifications is the protein completely functional. As we discussed earlier, a protein's shape is critical to its function. For example, an enzyme can bind to a specific substrate at an active site. If this active site is altered because of local changes or changes in overall protein structure, the enzyme may be unable to bind to the substrate. To understand how the protein gets its final shape or conformation, we need to understand the 4 levels of protein structure: primary, secondary, tertiary, and quaternary. Amino acid's unique sequence in a polypeptide chain is its primary structure. For example, the pancreatic hormone insulin has 2 polypeptide chains, A and B, and they are linked together by disulfide bonds. The N-terminal amino acid of the A chain is glycine, whereas the C-terminal amino acid is asparagine. The gene encoding the protein ultimately determines the unique sequence for every protein. A change in nucleotide sequence of the gene's coding region may lead to adding a different amino acid to the growing polypeptide chain, causing a change in protein structure and function. In sickle cell anemia, the hemoglobin beta chain has a single amino acid substitution, causing a change in protein structure and function. Specifically, valine in the beta chain substitutes the amino acid glutamic. What is most remarkable to consider is that a hemoglobin molecule is comprised of 2 alpha and 2 beta chains that each consist of about 150 amino acids. The molecule therefore has about 600 amino acids. The structural difference between a normal hemoglobin molecule and a sickle cell molecule, which dramatically decreases life expectancy, is a single amino acid of the 600. What is even more remarkable is that 3 nucleotides each encode those 600 amino acids, and a single base change, point mutation, 1 in 1800 bases, causes the mutation. Because of this change of 1 amino acid in the chain, hemoglobin molecules form long fibers that distort the biconcave or disc-shaped red blood cells and causes them to assume a crescent or sickle shape, which clogs blood vessels. This can lead to myriad serious health problems such as breathlessness, dizziness, headaches, and abdominal pain for those affected by this disease. William Warwick Cardozo showed that sickle cell anemia is an inherited disorder, meaning that the difference in the specific gene's encoding region is passed down from parents to children. As you will learn in the genetics unit, the inheritance of such traits is determined by a combination of genes from both parents, and these very small differences can have significant impacts on organisms. The local folding of the polypeptide in some regions gives rise to the secondary structure of the protein. The most common are the alpha helix and beta pleated sheet structures. Both structures are held in shape by hydrogen bonds. The hydrogen bonds form between the oxygen atom in the carbonyl group in one amino acid and another amino acid that is 4 amino acids farther along the chain. Every helical turn in an alpha helix has 3.6 amino acid residues. The polypeptides R groups, the variant groups, protrude out from the alpha helix chain. In the beta-pleated sheet, hydrogen bonding between atoms on the polypeptide chain's backbone form the pleats. The R groups are attached to the carbons and extend above and below the pleats folds. The pleated segments align parallel or antiparallel to each other, and hydrogen bonds form between the partially positive hydrogen atom in the amino group and the partially negative oxygen atom in the peptide backbone's carbonyl group. The alpha helix and beta-pleated sheet structures are in most globular and fibrous proteins, and they play an important structural role. The polypeptide's unique three-dimensional structure is its tertiary structure. This structure is in part due to chemical interactions at work on the polypeptide chain. Primarily, the interactions among R groups create the protein's complex three-dimensional tertiary structure. The nature of the R groups in the amino acids involved can counteract forming the hydrogen bonds we described for standard secondary structures. For example, R groups with like charges repel each other, and those with unlike charges are attracted to each other, ionic bonds. When protein folding takes place, the nonpolar amino acids hydrophobic R groups lie in the protein's interior, whereas the hydrophilic R groups lie on the outside. Scientists also call the former interaction types hydrophobic interactions. Interaction between cysteine side chains forms disulfide linkages in the presence of oxygen, the only covalent bond that forms during protein folding. All these interactions, weak and strong, determine the protein's final three-dimensional shape. When a protein loses its three-dimensional shape, it may no longer be functional. In nature, some proteins form from several polypeptides, or subunits, and the interaction of these subunits forms the quaternary structure. Weak interactions between the subunits help to stabilize the overall structure. For example, insulin, a globular protein, has a combination of hydrogen and disulfide bonds that cause it to mostly clump into a ball shape. Insulin starts out as a single polypeptide and loses some internal sequences in the presence of post-translational modification after forming the disulfide linkages that hold the remaining chains together. Silk, a fibrous protein, however, has a beta-pleated sheet structure that is the result of hydrogen bonding between different chains. Each protein has its own unique sequence and shape that chemical interactions hold together. If the protein is subject to changes in temperature, pH, or exposure to chemicals, the protein structure may change, losing its shape without losing its primary sequence in what scientists call denaturation. Denaturation is often reversible because the polypeptide's primary structure is conserved in the process if the denaturing agent is removed, allowing the protein to resume its function. Sometimes denaturation is irreversible, leading to loss of function. One example of irreversible protein denaturation is frying an egg. The albumin protein in the liquid egg white denatures when placed in a hot pan. Not all proteins denature at high temperatures. For instance, bacteria that survive in hot springs have proteins that function at temperatures close to boiling. The stomach is also very acidic, has a low pH, and denatures proteins as part of the digestion process. However, the stomach's digestive enzymes retain their activity under these conditions. We will conclude this lesson by stating that protein folding is critical to its function. Scientists originally thought that the proteins themselves were responsible for the folding process. Only recently, researchers discovered that often they receive assistance in the folding process from protein helpers, or chaperones that associate with the target protein during the folding process. They act by preventing polypeptide aggregation that comprise the complete protein structure, and they disassociate from the protein once the target protein is folded. See you in the next lesson.